Well, hello, this is Eric Topol, and for this edition of Ground Truths, I'm so delighted to have with me Professor Tony Wyss-Coray of Stanford, distinguished professor at Stanford, and directs the NIGHT Initiative for Brain Resilience. So, welcome, Tony.
Thank you. Thank you for having me, Eric.
Well, I've been following your career and your work for decades, I have to say, and what you just published a couple of weeks ago in Nature, the cover paper about internal organs, it blew me away. I mean, it's built on a foundation of extraordinary work. I thought we could start with that because to me, that's really a breakthrough in that when we think of aging and how to gauge a person's aging, things like the Horvath clock of methylation or telomeres are kind of not at all specific to any part of the body, just overall. But you published an extraordinary work about plasma proteins for 11 organs that uh, predicted the outcomes, things like heart failure and Alzheimer's. So maybe you could tell us about this. Seems to be a big deal to me.
Thank you so much. I'm honored. Really, I think if you work on this stuff, especially for several years, it feels sort of obvious to do it. Um, but I think, it is in a way, it is pretty simple. So what we argued is that the thousands of proteins that, are present in our blood, they must originate from somewhere. Now, a lot of proteins are, produced by cells throughout the body, but some proteins are very specifically produced, for example, only in the brain, or only in the liver, or only in the heart, because they have specialized functions. And we have, you know, been taking advantage of that in clinical medicine, where you measure often, one of these proteins to sort of diagnose pathology in a tissue. But we took this just a level further and said, well, let's just find out of thousands of proteins that we can measure, assign them to specific organs and tissues, and then see whether they change with age. And many of them turn out to change. We found, about 1,500 proteins or so in the study that we did, although that number can grow dramatically if we, keep improving our technologies or techniques to measure them. And many of them come from the brain or from other tissues. And because they change with age, they tell us something about the aging of that organ. And as others have shown in the field, including Steve Horvath, is that prediction of the age, if it doesn't really match exactly your actual age, contains information about the state, the physiological state, or the risk to develop organ-specific disease.
Right. And you found that about 1 in 5 people had evidence of accelerated aging of one organ, which, of course, is really starting to nail down the ability to detect aging, to relocalize it. And what strikes me, Tony, is that now because we're seem at the cusp of advancing in the science of aging, a field that you have done so much to propel forward. And one of the issues has been, well, how are we going to prove it? We can't wait for 20 years to show that whatever intervention led to promotion of healthy aging. But when you have a marker like this of organ specificity, it seems like the chances of being able to show that intervention makes a difference is enhanced. Would you say so?
Yeah, absolutely. I think that's one of the most exciting aspects of this, that we can now start looking at interventions, whether they are, a specific interventions that tries to target the aging process, or, just a drug, let's say a cholesterol lowering drug, or blood pressure lowering drug. Does that have a beneficial effect on the heart, for example, on the kidney, or, and you can also start thinking of lifestyle interventions, whether they actually have an effect, right? If you started exercising, you collect your blood before, and then a year after you have an exercise regimen, does that actually change the the age that we can measure with these different clocks?
Right. Well, I mean, it's really a striking advance in biomarker of aging. So that gets me to work you've done well over 10 years, which is that you could identify uh, that giving young blood, first, of course, in mice and then later verified in people, could improve cognitive function. Um, and in older, whether it's experimental models or people. So, what are your thoughts about that? Is that if you, that's something you've been ruminating on for many years, and I'm sure there are places around the world that are trying to do this sort of thing. What do you think of that potential?
Yeah, so and they're really this recent observation or study really came out of, that finding that young blood can change the age of different organs. And, we were not the first to show this. We showed it for the brain, but Tom Rando, who studied muscle stem cell aging, showed this, a few years earlier in the muscle. And then we worked with Tom to explore this for the brain. But it shows sort of that this, the composition of the blood is really not just reflecting the age of organs and tissues, but it actually also affects them, it directs them in a way. And so you can speculate that, if you had an organ that shows accelerated aging, because some of the factors end up in the blood, they might actually induce aging in other tissues and so promote the aging process. And people in the field have also shown that this is true for specific cells. We call them senescent cells. So these are a specific type of cell that seem to somehow stop dividing and assume the state that releases inflammatory factors. These cells too, they seem to almost infect the neighborhood where they live in with an age promoting sort of um, secretome, we call it. So they release factors that seem to promote aging locally, but potentially across the organism. And interfering in that could potentially have rejuvenating effects. And so that brings us back to this observation that young blood could potentially rejuvenate organs. We know old blood can accelerate it at least in mice, right? So could we neutralize the age promoting factors in people, and could we, deliver sort of the rejuvenating factors? Now, what's been frustrating for me is that it has been incredibly challenging to identify the key factors. And I think we became to realize as a field that there's not one factor, there's not one magic factor that will keep us young or keep our organs young, but rather different cells and different organs in our body seem to respond in different ways actually to this young blood. We can show this with molecular tools. We can show that every cell actually responds. So if you take a mouse, an old mouse, and you give it young blood, every cell in that mouse shows a transcriptional response to the young blood. But some of them, may regenerate mitochondria and others activate other pathways. We see that stem cells respond particularly well, the stem cells of the immune system, hematopoietic stem cells, while other cells show less of a response. And that to me suggests that they respond to different factors in the young blood, and that, they have very specific receptors probably that recognize some of these beneficial factors and then respond in a specific way. So what we need to figure out, I think, as a field to translate this really to the clinic is what are the key factors, and will it be possible to make a cocktail that sort of mimics nature's, elixir, if you will. And, I said this before. It's almost like the fountain of youth is within us, but it just dries out as we get older. And if we could figure out what are the key factors that make up this fountain, we could potentially, either as a treatment, deliver it again or reactivate that fountain so that the body produces these factors again.
Well, that's something that years ago I was very skeptical about. And because of your work and others in the field, I've come a long way thinking that we're on the cusp of really identifying ways to truly promote healthy aging. And so this is really, extraordinary time in our lives. I wonder, you of course mentioned two critical paths that have been identified: the senescent cells, removing them, or the infusion of young plasma, young blood. And would you say it's too simplistic to reduce this to decreasing inflammation? Is that really the theme here, or is it much more involved than that?
I think inflammation has a big part in that, but inflammation is such a broad term and such an ill-defined term
Yeah. Yeah.
that I can say yes to your question, and I'm probably not going to be wrong. Um, but if we really want to know, which molecular pathways in the inflammatory cascade are key to this, detrimental process that seems to accelerate aging. I think we have to work a bit harder and really define what we're saying. You can't just have thousands of proteins or genes that have something to do with immune and inflammatory processes called inflammation. Then yes, everything is inflammation. But I think we have to be more precise, otherwise you can't really target it. Having said that, if we use sort of the conventional tools that biologists use, these pathway analyses, if we give young plasma to an aged organism, then the top pathway or one of the top pathways in almost every cell is inflammation, suggesting that we reduce the inflammatory process. But again, it's in a very broad sense, and I want to know more what we're finding. In fact, one of our first observations when Saul Vileda was in my lab and did these first parabiosis studies to look at factors that might promote brain aging, he identified beta-2 microglobulin and eotaxin. Eotaxin is a chemokine that is involved in a lot of, sort of inflammatory responses and has actually more recently again, been implicated by Michel Monge here at Stanford to be a mediator of, the chemobrain, as people call it, at least in animal models. And we showed that it's part of the aged plasma that causes sort of an acute impairment of cognitive function in mice. So that would be an example of a bad factor. And that is part of an inflammatory cascade, but we want to know what exactly is it. We tried a small molecule that targets the receptor, one of the main receptors for this chemokine, but unfortunately in humans, but unfortunately that compound had some side effects on the liver, and we never got to really test the question: is this, potentially important? It's one of the challenges of, drug development, that you often don't get to test your questions because the drug has side effects that don't allow you to do that.
Well, speaking of drugs out there, this past week there was a very provocative paper from Daniel Drucker, University of Toronto, on the GLP-1 effects on brain inflammation. And interestingly, you may have seen it, but with mice that were either knocked out of GLP-1 for their blood cells or their brain, it was clear that inflammation reduction with these drugs, and they tested several different GLP-1 agonists, worked through the brain, which was really fascinating. And I wonder, Of course, these drugs are now, they craze for anti-obesity. But do you see something like that, this peptide agonist, as a potential way to achieve some of the effects that you've been working on for a long time?
Yeah, I think this is extremely fascinating. I mean, these drugs, we don't understand them exactly what they're doing, as you know, for many drugs, but it's really amazing the effects that you see. And you know, I'm very hopeful. There's a large phase 3 trial in Alzheimer's disease ongoing. So, and the phase 2 looked very positive, very promising. So, you know, it is really possible that th that there are key pathways that are responsible for, you know, cognitive decline and cognitive impairment, and that inflammation is a key aspect of that. Again, inflammation in a broad term, we need to define it, but it could be that it goes through, you know, through these GLP receptors, um,
Yeah,
and
yeah.
that
Well,
is might be some regulator of a broader process. But you know, we see, for example, with aging, just with normal aging, you get activation of inflammatory pathways in the brain vasculature, and young plasma reduces these changes acutely. And maybe this is, you know, all part of a um, just dampening that inflammation gives you some additional, you know, brain power, if you will, lack of a better word. And that much of, you know, at least the early stages of cognitive impairment that lead to Alzheimer's disease are relatively transient and are more like a fog, like we say, you know, the chemo fog, the chemo brain or brain fog, but that, you know, with COVID that you also commented on very prominently. That that suggests that it's not a structural damage early on, but that it might be some soluble factors that would go a long way if you could just suppress them.
right, right. Well, it's really fascinating to see, and I'm glad you mentioned the phase 3 trial in Alzheimer's for this one class because I think that's expected in 2025 to read out, and that'll be really important. But I wanted to ask you because now there's many shots on goal to change the natural arc of aging. And all these companies like Altos and Unity and Calico, and I mean, there's so many of them I can't even keep track. They're all taking different strategies. I have to think because they need to have their own intellectual property. What do you see as the alluring ways that we're going to be able to modulate this process?
That's a very tough question. Um, I think it's hard to predict, I would say, you know, like always in biology. First of all, as you know, what we discussed earlier, it could be that a drug that tries to test the pathway, like, you know, one that Unity tried, has side effects, and, you know, you can't actually test your hypothesis. Uh, but I think one of the key sort of aspects of the aging process is really that it's global across the organism, but it's also very localized. And so it's possible that targeting the aging process will first show benefits in individual tissues. If we target, you know, the aging process in one particular tissue, that might show the first benefits. But then again, it could be that if there is sort of a key inflammatory driver that you know is to some extent responsible for overall aging of the organism, and you manage to target that and slow it or block it, you may have an organism-wide effect.
Hmm.
But I
Yeah.
think we have to be realistic that, you know, this is going to be an incremental process, I think.
So, is there anything that you've seen that has grabbed you as having tremendous potential that is new, or is it really, the things that have already been percolating that we know about?
Yeah, I mean, the GLP-1 study, I've been actually a bit involved in that. I find this really fascinating.
Yeah.
Um, yeah,
Well, that
I
I
mean,
spent
not in that study that got published, but in sort of more on the cognitive side.
right, right. I thought that was especially welcome news because the drugs we have now for Alzheimer's seem to have, some pretty serious side effects and somewhat low efficacy relative to the risk assessment. So this would be a drug that we know now as people have taken for years. That I did want to get back to you on with you on the durability. So if you give young blood to an old person who has, let's say, mild cognitive impairment, will you see a durable impact, or is it just a very short-lived one?
I think some of the effects will be durable, and I'm saying that because of an experiment that James White and Vadim Gladyshev did. They used this parabiosis model where you suture a young and an old mouse together. They left these mice together for 2 months, I believe, or 3 months, and then they separated them and let them live. And looked at how long does an old mouse live that was paired with a young mouse for a few months compared to an old mouse that was paired with another old mouse. And they saw that there is clear extension of lifespan if the mouse was exposed to young blood. Now, this is in the context of, two major surgeries, first suturing them together and then taking them apart. But, I always note that when I present this experiment, but I also say at the same time that this is the problem that a lot of older people have, right? When they have a surgery, they don't recover from it as well. And it's often the beginning of cognitive decline. If you ask families, when did it start? Oh, they had heart surgery, or they fell and had hip surgery, or something like that, or a major infection. Zoster or something like that. That is often the trigger where the it's almost like, the organism is hanging in there and is still functioning, and then there's an injury and it collapses. And so, what's remarkable with the rejuvenating intervention with parabiosis is that it seems to overcome this to a very significant event. And they also showed, with many other tools, including with the Horvath clock, that tissues are actually getting younger um, through this process. We have also found that, stem cells are rejuvenated for a long period of time if you treat with young plasma infusions in mice. And so I'm hopeful that some of the effects are going to be long lasting, but you know, practically you would probably still treat people on a regular basis, right? Like we do with all drugs, but maybe you would do an infusion every 3 months or every 6 months. And we're still trying with a company that I started, um, Alcahest, to convince people to do a phase 3 clinical trial and see how far we can push this.
Well, it'd be really interesting to see you get that done. Then going back to the senescent cells, which is another leading prospect, it seems to be more difficult to get these cells out of the body. We know they're bad actors, but it isn't like we can, very selectively remove them. But what are your thoughts about that approach?
Yeah, I mean, I'm always really puzzled and amazed at the effects that people show with, senescent cell removal in animal models, right? Um, there is something really almost magical there that you remove these few cells and, the body is doing much better. Um, so I think, we should keep trying very hard to translate this to humans. But it's possible that, again, there very likely different types of senescent cells in different tissues. I mean, in the brain, there are no rapidly dividing cells, so it's not the classic, arrest of cell cycle, but it's probably more like an astrocyte type of cell that might mimic a senescent state. But I think it will be, very much organ specific, and may require very specific interactions or drug targets, and drugs.
Right. Well, then gets us back to kind of where we started. Before your what I consider a landmark paper, um, it would be difficult to be able to go to a regulatory body like the FDA and say, we show that this is affecting the aging process. And we show in, 3 organs, 5 organs, whatever the 11 organs you could track, we are reversing the aging process. I mean, you have that now as a extraordinary finding. Do you think that will help accelerate the field by having not having to have a whole body aging story with a epigenetic clock, but rather, a much more pinpointing organs that can be helped, that they can be promoting healthy aging? It seems to me this is where not only advancing the theories of how to do it, but the proof that you have done it. It seems like this is what, why I consider it such a extraordinary finding.
I totally agree with you, but I'm a bit biased. I think, this is what we need. I mean, this was always a criticism to me, and we're very good friends with Steve Horvath and Morgan Levine, who came up with these remarkable aging clocks, Ken Raj, right? And but my question was always, how can you get information about the whole body aging by looking at changes in blood cells or cheek cells, right? That cannot contain information about how your pancreas ages or your heart ages at a high resolution. It will correlate, admittedly, but it will not give you tissue-specific information, most likely. So, going more directly at molecules that are derived from cells across the organism is, of course, going to be much more informative. We've just started to do this. It's a concept. Um, we are super excited about, looking now at large data sets like the UK Biobank. We just get access through a collaboration to 50,000 individuals, where we have 1,500 protein measurements with a different proteomic platform. And it seems most of the findings replicate. So you see a similar, very strong risk for people who have older brains to develop dementia in the future. And we still see these extreme organ agers that I find very puzzling.
Yeah, no, it's really striking, and the fact that you could replicate the best biomarker for the brain, the tau 181, through these proteins is exceptional. How much did machine learning AI help you in deciphering this large data set of proteins? Was it really critical, or was it just a small part?
Oh, it's certainly. I mean, this is. I think the terminology. I'm not so clear, and I have to admit, I'm not a computer scientist by any stretch. Um, but I think this is classic machine learning, statistical elements of learning. How Rob Tibshirani described it. So we use still ENet. We use linear modeling uh, in these models. Um, but I think it will become more sophisticated, and I think AI will help us to bring this to a much higher level by basically learning from the relationship between proteins directly and then compare that in healthy people versus control. Similar to what Christina Theodoris recently did for gene expression at the single cell level. I think we will see that, and we're trying this, and I'm sure others are trying this too at the protein level.
Right, right. Well,
But
no,
the current study uses really more traditional machine learning models that, are sophisticated, but it's not, you know, I'm not sure we call this artificial intelligence.
you sure? Well, I think, as you say, it can build on that and, putting
Yeah,
in transformer models to more data sets and where the future
yeah,
goes, you'll get even more precision output. Well, Tony,
absolutely.
this has been a real joy. I have to say, congratulations to you and your team for such
Thank you.
exceptional work. This has been a multi-decade, one layer of after another of building on the science of aging, particularly the brain aging. I've learned so much from you, and you. I have to say, the paper you just published, you and your group, got me excited. I mean, I really thought of all the things I've seen on aging, this was the one that really opens it up for all the other possible ways to claim it's you're making a difference. You've got a way to you have a metric that's emerging, and so kudos to you. And I know this is got to be, of course, that you probably just think, oh, it's just one more thing we've been doing, but I'm so duly impressed.
Thank you so much. Thank you for the nice word. It means a lot.
Well, keep up the great stuff because we're all depending on you so that we can have a better arc of our healthy aging process, and we'll keep in touch.
Thank you. Thanks so much.